so I'm sure you guys are all feeling like this, either because it's the last section of NST 103 or it's an election. Which one is making you feel like this? But to just back up a little bit about where we've come from and where we're going. So in the first section, recall that we talked about digestion, absorption of carbohydrates and lipids. In the second two sections, which were taught by Professor Hasek's school, you did intermediary metabolism and protein metabolism. And I just want to mention that for any questions related to this, these sections direct them towards Professor Sewell or towards the GSIs, just to avoid any kinds of confusion since I wasn't present for those classes. And then we're moving on to the fourth section and the last section. So this will take us through November. So we'll talk about iron and heme over the course of this lecture and the lecture on Monday. And then we'll move into select vitamins and minerals for the remaining 4 lectures. And so we end by the end of November, go into the reading and study week. And I think our final is until December 16th, so tons of time to study and know all the material by the time the final comes around.
Thank God.
Okay, so and as a reminder, I'm going to have 2 office hours, one on Friday, 11 to 12, and one on Tuesday, 3 to 4. And then Clark and Pete will keep having the same office hours. I think these are still correct. Pete has office hours Tuesdays, 10 to 11. Clark has office hours 3 to 4 on Thursdays. And the same with the review sessions. So no review sessions this week since we only have one lecture, but they'll start out having review sessions again next week on Thursday and Friday. Okay, so we're going to move on to talking about iron metabolism. So concerning the biology of iron in man. Now I want to stress iron in man because unfortunately we're not studying the biology of Iron Man, which that would be pretty cool since he's a member of the Avengers and defender of the world. But Iron Man does require iron in his biology like the rest of us too. So we're going to be studying iron in man, which is a trace element, transporter of electrons, carrier of oxygen, defender of our blood cells. So we'll talk about the relationship of iron in our body, what we use it for, why we need it, and why it's important, particularly for our blood cells. So today we're going to go through kind of some of the basics about iron, talk about basic properties of iron and why it's useful, talk about the flux of iron in our body and the different amounts of iron and where it's stored in our body. Then we'll talk about intake and absorption, its circulation through our body, and then regulation of these various iron pools. Okay, so iron is an essential trace element, so this is a little bit different from some of the things we were talking about at the beginning of the semester, which were more macromolecular nutrients that were used for biomass or used for breakdown gain energy. This is an essential trace element that's utilized by a lot of enzymes or proteins in our body. And it's useful because of its chemical properties. So for example, it's able to form bonds with electronegative elements, such as oxygen. So this is going to be really important for transport of oxygen through our body. So this is how hemoglobin and heme bind oxygen and aid in the movement of oxygen through our body. And iron's also really useful because it functions well in redox reactions. So it's able to accept and donate electrons. So for example, we can see in our body we have iron in mostly 2 forms, H3 plus and H2 plus. And when we add an electron to H3 plus, we convert it to this reduced form, the H2 plus, right? And so this can go in either direction, and so it can donate and accept electrons. And this is going to be important for movement of electrons or transport of electrons, for example, in electron transport chain. And these different forms of iron have different names and nomenclatures. Sometimes you'll see this as Fe3 with this Roman numeral, or it will be referred to as ferric iron. And so ferrous is Latin for iron, and you'll see a lot of these enzymes have this kind of name, ferrous, or have that present in their nomenclature. And then the Fe2 plus is referred to as ferrous iron. So ferric is Fe3 plus, the oxidized form, and then ferrous, Fe2 plus, this reduced form. And we'll use these interchangeably. Okay, so it's essential for a lot of biological reactions and processes. One I just mentioned was oxygen transport and retention. So things like hemoglobin or myoglobin coordinate a heme molecule, which is able to bind the oxygen. And in the case of hemoglobin, it'll be high in erythrocytes and aid in movement through the body, or for myoglobin that aids in retention of oxygen that can be used by our muscles. Iron is also present in a lot of enzymes in our body, so it's important, for example, for ribonucleotide reductase, which reduces ribonucleotides to deoxyribonucleotides. So this is going to be important for DNA synthesis. This is just an example, but a lot of enzymes have a coordinated iron present in the enzyme. And it's critical for electron transport. So there are these iron-sulfur-containing proteins, as well as heme-containing proteins that function in the mitochondrial electron transport chain and function moving electrons through the different complexes in the mitochondria. And so by contributing to these diverse types of processes, iron is going to be important for a lot of different intermediary metabolism and a lot of physiologically important reactions in our body. Strikingly, though, free iron can have very toxic effects for the cell. And so redox cycling between the ferrous and the ferric forms of iron can generate reactive oxygen species, which can be very harmful to the cell. And this is primarily through this Fenton's reaction. So here what we see is there's a hydrogen peroxide, and as we gains an electron from ferrous iron, and this leaves as the ferric form, and this splits into a hydroxide and a hydroxyl radical. So these can go on and non-specifically modify various macromolecules in the cell and damage the cell. So it can pretty much modify and react and damage things like lipids, carbohydrates, nucleotides, and amino acids. So this has the potential to be very toxic. Right? So we have this balance of iron's important for biology, but it can also be toxic. And there's even a form of cell death that's iron dependent, and it's referred to as ferroptosis. And this was discovered by a friend of mine, Scott Dixon, a few years ago. And this is a form of iron dependent non-apoptotic cell death. So it's distinct from apoptosis, and it requires iron. And what he found was that this ferroptosis, the cells die because of lipid peroxidation, lipid damage. So you can block it with iron chelators, or you can block it with antioxidants. Okay, so iron has the potential to be quite toxic to our cells. So iron's important for our cells, but it can also be toxic. So how do we kind of get around these conflicting properties of iron? Right? It's useful, but potentially toxic. And so the way the body does that is it constrains the iron reactivity by associating iron with different molecules within the protein, prosthetic groups. And there are a few that are important for us. There's heme, and so here we see the heme structure, and we'll talk about heme synthesis and degradation on Monday. So that's a very characteristic ring structure, and you see this central iron coordinated by these nitrogens within the heme ring. And this heme ring is further associating here the histidine within a protein and the molecular oxygen. So this probably is at the heme present hemoglobin that's oxygenated and moving oxygen through our body. Okay? And so here the iron wouldn't be toxic, it's coordinated within a heme and wouldn't be capable of performing that Fenton's reaction, producing these reactive oxygen species. Similarly, we can have iron that's coordinated by sulfurs. So these are referred to as these iron-sulfur clusters. And these mostly come in two flavors, the 2 iron, 2 sulfurs. So here we have 2 iron and these 2 central sulfurs. There are actually more sulfurs than that because each of the irons is coordinated by cysteine residues, which contain sulfur. Okay, so we have two cysteines here coordinating this iron, 2 cysteines here coordinating the iron, and 2 central sulfurs. Okay, and then another example is the 4 iron, 4 sulfur. So here we have 4 irons and 4 sulfurs, and then we have additional coordination by these cysteines contributed by the protein. Okay, so this is just another example of how iron can be incorporated into a protein and coordinated safely to prevent this reactivity. And these iron-sulfur cluster proteins are important within electron transport in mitochondria, and they're also going to be important for iron sensing. So we'll talk about a specific example of this in which a protein that contains an iron-sulfur cluster senses the concentrations of iron in the cell, and in this way can monitor and regulate the biology of the cell in response to fluctuating concentrations of iron. And that will be dependent on its iron-sulfur core. We'll talk about that towards the end of today's lecture. And then the other form that can be safely stored is within ferritin. And this is a large iron storage compartment. And it has this large luminal space in which it can store iron in the ferric form. So in this form, it can store up to 4,000 or 5,000 molecules of iron. So it's a very dense storage form. And then this will also prevent reactivity of the iron. Okay, so in terms of total body iron, is about 4 grams in our body. This will vary depending on your size and your diet. And we can think of it as being divided between 3 major classes or groups. Okay? So there's the functional iron. So this is iron that's present within a protein that might be playing a role in oxygen transport or electron transport. We refer to that as functional iron because it's playing this important role. So some examples are the iron in hemoglobin, the iron in myoglobin. So both of these would be heme-containing proteins that are storing or moving oxygen, or the iron in these iron-containing enzymes, right, like in the electron transport chain. So these we refer to as functional iron. That iron is playing an important role in the function of these proteins. We have storage iron, and this is mainly in the form of ferritin, so locked in that giant sphere. And this is mostly in the liver, in muscles, and in macrophages. Okay, so these are kind of high iron tissues. And then we have a small amount of iron that's in the form of transport iron. So it's fluxing between these various pathways, between functional iron and between storage iron. So this is really small amounts that are present in our plasma, in our blood, and it's bound to a transport protein called transferrin. So this is a different representation of kind of what I just said. So we have within our body around 3 to 4 grams of iron. This is going to be distributed between the functional class, the storage class, and the transport class. And most of the iron in our body is present in erythrocytes, so in red blood cells. So these have around 2.5 grams of iron. So total we have 3 to 4, and 2.5 is in the red blood cells. So the majority of iron is going to be present in our red blood cells, and heme coordinated by that hemoglobin. And recall that these are very long-lived cells, right? Around 120 days. Okay, we also have a large portion of iron stored in those ferritin cages, so around a gram of iron will be present in the storage compartments, ferritin. And this is going to be important to provide a buffer. So if we have too much iron, we can store more in ferritin. If we don't have enough, we can free up iron from those ferritin storage compartments. And then about 300 mg is present in that functional class, so present in proteins and myoglobin and other enzymes. Okay. So most present in the red blood cells, some in storage, and then a smaller amount in various enzymes and proteins. And then a very small amount, 4 mg, is present in our plasma. So this is circulating through our body, and this is how we're moving and transporting iron through our body. And you can see that there are some arrows going back and forth here. So about 5 mg a day fluxes into this plasma from our storage compartments or from proteins or to proteins. And about 20 mg a day fluxes into this transport compartment from the red blood cells. So this is usually due to breakdown of old red blood cells. Okay? So usually these are really long-lived, but as they're coming to the end of their life, the body is really good at recycling the components and recycling that iron that's present in those erythrocytes. So they're going to be broken down in macrophages, releasing the iron, and then this can be resynthesized into heme and packaged back into new red blood cells. So the body's really good at recycling the iron. So in addition, this plasma iron levels are going to be affected by influx from our diet as well as excretion. So we lose about 1 to 2 mg a day of iron But you can see that this is mainly lost by loss of cells. So we slough some GI cells, we slough some skin, and then a tiny amount is lost in the urine. So our body is not really actively excreting iron. This is just kind of some iron lost with lost cells. And we could lose a lot now if we, for example, have a wound and we lose blood, right? Because the majority of our iron is stored as blood. So if you lose a lot of blood, or you can have a big deficit in the iron. That'd be the fastest way to become iron deficient. So we lose about 1 to 2 mg a day through the loss of some of these different cells. And so we have to balance this by absorbing a similar amount, right? 1 to 2 mg a day. And so we get this from our diet. Right? And so if we have long-term changes in what we absorb, so if we're absorbing too much or if we're getting not enough iron from our diet, that'll eventually be reflected in our iron stores, either as a deficiency or as an overload iron. So just to reiterate, the body contains about 4 grams of iron. All of our cells contain some iron, mostly complexed with these various metabolic proteins. But we have specific tissues which are high in iron, either because they require iron for their function or because they're iron storage cell types or tissues. Right? So erythrocytes have really high levels of iron because they pack in the hemoglobin for oxygen transport. Right? So they're using that iron for their function. So 2 to 3 grams of iron. And then there's also a lot of iron in liver, so liver has a lot of ferritin, will function in storage of iron. The spleen and macrophages are important for the breakdown and synthesis of heme and the recycling of these blood cells. And then muscle has myoglobin, which has heme in it and stores oxygen for our muscle use. So these will have perhaps higher levels of iron compared to other cell types. And then about 4 grams is complex with transport proteins in our blood or in our plasma for distribution. So this is a really small amount of iron that's in our blood, but there's a lot of flux in this compartment. So every few hours, the plasma in our blood turns over because there's constant transport to different tissues. It's constantly gaining new iron from either our diet, from our stores, or from breakdown blood cells, and delivering that iron to other tissues. So there's a lot of flux through this compartment. So in terms of iron deficiency and iron overload, there are a lot of health problems associated with both. And iron deficiency is probably the most common nutritional deficiency. And it's the most common cause of anemia. And so we'll focus on the relationship of iron and anemia on Monday when we talk about blood cells. So we'll focus on this in a lot more detail. Any questions so far about that? It's pretty straightforward. Okay. So in terms of iron absorption, there are 3 general factors to keep in mind when we think about iron absorption from our diet. Of course, the most obvious is the iron content of the diet, so how much iron is actually present in the food we're ingesting. Maybe less obvious is the bioavailability of that iron, so various food matrices are going to affect the solubility of the iron, the pH is going to affect this, and the form of the iron can also affect solubility and absorption. We'll talk about that a little more. And then finally, the capacity of the enterocytes to absorb the iron. So there can be changes in the expression of transporters and iron exporters in the enterocytes, which will impact our ability to efficiently absorb iron from our diet. Okay, so we consume about 10 to 20 mg of iron per day. This is all kind of a general for Western diets. So we consume about 10 to 20 mg of iron per day, but we only absorb about 1 to 2 mg. You can really see already that the absorption is not completely efficient. There's going to be a lot of effects of bioavailability. So in the US, for the diet, about 2/3 of that iron is consumed as ferric iron, so that's the Fe3+, the oxidized version, and that's mostly coming from vegetables. About 1/3 is consumed as heme iron. So that's going to be coming from meat and the myoglobin. So myoglobin in the muscle contains heme. But heme iron is much more efficiently absorbed. So about 2/3 of our body iron is going to be derived from heme iron. So even though we're consuming more iron from our vegetables, majority of the iron is coming from the muscle because it's more efficiently absorbed in this heme form. That's something to consider when you modulate your diet. That just having more iron, for example, in vegetables, doesn't necessarily mean you're going to absorb all that iron.
Can you have supplements that have heme iron, or are they mostly ferric iron?
So the supplements are ferrous iron, and they usually come in the form of ferrous sulfate or ferrous fumarate, and those are you're able to absorb those pretty well. I don't think there's ever heme iron. It's probably because it'd be really difficult to obtain or to synthesize. And so usually you get it as sort of ferrous conjugated with fumarate or sulfate.
And that's be absorbed like similar to heme iron?
The efficiency of absorption is a little bit different, but it's very effective.
Okay.
And that would be the form that you would get in fortified cereals and flours. So a lot of cereals are fortified with iron as well, which can be a very good source. And that will be easier as the ferrous sulfate or fumarate form.
So is heme iron ferrous iron or ferric iron?
It's ferrous, but it's bound within that porphyrin ring. So it's the effect of that porphyrin ring has a large influence on its solubility as well as its absorption routes and how it's metabolized. But it'll be a ferrous Fe2.
And if it's not bound within the heme, like that it's not associated, it would be toxic. The ferrous.
They can't get out of tooth.
In the cells, yes.
No, but the ferric wouldn't be. Like the ferric.
The ferric is so in free form that would be toxic. So the cell usually stores that really rapidly once it gets in the cells. We'll talk about that. So we try to minimize free iron in our cells. So we have these two different forms that are going to be present in our diet, three different forms here. The heme, either present in heme, so the iron's coordinated in the center of this heme molecule, or the non-heme, which can be either ferric or the ferrous form. And these are going to have really different properties and different routes of transport and absorption in our body. Okay, and just a reminder that this heme iron is mainly coming from myoglobin, so this is a really good source of highly absorbable iron. So myoglobin and hemoglobin are these related proteins. Hemoglobin has 4 subunits that are very similar to myoglobin. So it has 4 coordinated heme molecules, whereas myoglobin has a single subunit and a single heme that's coordinating a single iron. And so when you ingest meat or meat products, you'll break down the proteins in your gut and release the heme for absorption. Again, so there are several factors that can influence iron solubility and absorption. One is the pH. So acidification, like in your stomach, will increase the absorption and the solubility of non-heme iron. So both ferric and ferrous become more soluble as you acidify the pH. Something like ascorbic acid, vitamin C, can actually reduce ferric iron to ferrous iron, and this increases the solubility and also the absorption. So we actually can't absorb ferric iron, so it has to be in the ferrous form. And so vitamin C has been shown to increase the reduction of ferric iron to ferrous iron, and both increase the solubility as well as the subsequent absorption by pterocytes. And then there are a variety of chelators, both solubilizing chelators or precipitating chelators. So these are just binders, molecules that bind the metal. And in terms of solubilizing chelators, things like amino acids combined to ferric iron and help maintain its solubility and so influence subsequent absorption. And then there are a variety of precipitating chelators, like phytates from plants, which could reduce the solubility of ferric iron and this would reduce subsequent absorption. So in this graph, we're just showing kind of this relationship between iron solubility and pH. So on the left here, we have the percent of iron in solution, and then on the bottom, we have pH. So if we just look at ferric iron, we can see that at very acidic pHs, it starts to become very soluble. But once we get to neutral pHs, it's very insoluble. You have to be soluble in order to be absorbed. Ferrous iron, so the Fe2+, has a similar relationship with pH, so it's more soluble as you get more acidic. But you can see that it's been shifted to the right. So in the neutral pH, ferrous iron is pretty soluble, and you can absorb ferrous iron. Okay? And then heme iron has the opposite relationship. It becomes less soluble as you become more acidic. But similar to ferrous iron, within this neutral pH, which is going to be present in your gut lumen, in the small intestine, it has a considerable solubility. This is going to contribute to its efficient absorption. So iron is absorbed in our small intestines by the enterocytes, our favorite absorptive cell. So here we have just the representation of a villus in our small intestine. We have our enterocytes bordering the villus, and then we have this crypt, right, which is right next to the villus. And if you recall, the intestinal crypt stores stem cells, which will differentiate over time into the enterocytes. Right? So stem cells present in this intestinal crypt. And there's an interesting relationship between these stem cells and iron absorption. So as these stem cells differentiate, they start to express transporters on their surface, which allow them to take up iron from the gut. Okay? So when they're present here as stem cells, they're unable to absorb iron from the gut. Okay? And then as they differentiate, they express transporters and they can start to absorb iron from our diet. Right? This makes sense. And so while they're in the crypt, they can't absorb iron from the diet, and so they gain iron from the plasma, from our blood. So this is how all the rest of our cells in our body get their iron. They get it from the iron circulating in our blood. Okay, so they're absorbing iron from the blood, and then over time, as they differentiate, they lose the ability to absorb iron from the blood and start to gain iron from the gut lumen. This makes sense? Okay. And so why this is interesting is because while they're present in the crypt, they're able to sense the concentration of iron that's fluxing through our body, that's fluxing through circulation, because they're actually taking this iron up and losing it. Okay? And so if there are changes in the iron in the body, in the circulation, they'll be sensitive to this. And this will influence their expression of iron transporter proteins. For example, if there was low levels of iron in the circulation, they might start upregulating transporters so that they can absorb more iron. What's important to note is that it's really slow, its effects on absorption. So it might detect these changes in iron levels in our body, but it takes about 2 days to differentiate. So it'll change the ability of the body to absorb iron, but that change will be slow to happen, about 2 days. So this is just an overview of enterocyte iron uptake. So there are a few events to be aware of, and we'll go through each of these steps in detail. So as I mentioned, iron can be present in any of these different forms. It can be present as heme, coming from myoglobin and broken down muscle and meat, or it can be present as ferrous or ferric iron. These are going to have very different properties and different import methods. So these have to be imported across the apical membrane into the cell, cellular vents, and they'll be exported across the basolateral membrane into our circulation for transport. So for this first step, we express a particular transporter, a divalent metal transporter, on the basolateral membrane of our enterocytes. And this transporter can transport divalent metals, as the name kind of implies. So it'll be able to transport things like copper, zinc, and iron. So iron has to be in the Fe2 or the ferrous state, and it can be transported by this DMT1, this dimetal transporter 1. but it has to be in this ferrous state. So ferric iron our body cannot absorb. However, we do have an enzyme, this duodenal cytochrome B or DCYT, which can reduce ferric iron to ferrous iron. So it can add an electron, get ferrous iron, and then this can be absorbed. So ferric iron is a pretty challenging molecule. It has low solubility, it has to be really acidic for it to be soluble, and it has to be reduced to ferrous iron prior to absorption. And that's why a lot of that ferric iron that's coming from our plant diet often doesn't get absorbed. And then heme iron is absorbed very well. And this is thought to be, at least in some part, mediated by heme carrier protein 1. And heme carrier protein 1 definitely can transport heme into the cell, but it seems likely that there are other transporters. So if you get rid of heme carrier protein 1, the cells still seem to take up heme. So it's likely there are redundant transporters as well, in addition to heme carrier protein 1. But this protein can definitely transport heme. So I don't think we have the complete story in terms of heme transport in our cells. Okay, so after being transported into the cell, we have transport of the ferrous iron, right, through DMT1 into the cell. So we have this labile iron pool, and then we have transport of heme into the cell. This heme can be broken down to release the iron present in the heme by an enzyme called heme oxygenase. Okay, so it's going to break that heme ring. This is going to be important when we talk about heme degradation pathways. But for this lecture, it's just important to know that when you break that heme ring, you can release the iron that's coordinated by those nitrogens within the heme, and it can exit as ferrous iron and enter this labile iron pool. So we take up ferrous iron, or we get ferrous iron by breakdown of our heme. Right? So both of them are contributing to this labile transient iron pool. And this is really transient, so iron can be really toxic to our cells, as I mentioned. So it's transient, and usually this is kept at very low levels. So the sources are just going to be broken down heme, uptake from the diet, or release from storage pools. So right, this labile iron pool has a few different potential fates. So we want to keep this low, and so we can either store it, right, in ferritin. We can target it for utilization, so incorporation into newly synthesized heme-containing proteins or newly synthesized proteins containing those iron-sulfur clusters, or we can target it for export. Right? If we're in an enterocyte, maybe we want to export it into the circulation for use by the rest of the body. So we've taken up iron. It can be either stored, used, or exported. Yeah.
What does transient mean?
Transient just means it's very short-lived. So we're only going to have this labile pool for a very short time. So usually, as soon as it's taken up, it's usually targeted either for storage, utilization, or export. We don't want to keep it around for a long time in this free state because of its potential toxicity. Okay, and we mentioned ferritin in terms of iron storage. This is just a little bit additional information on ferritin. So it's a really unique protein that's evolved to store a ton of iron. So it's made of 24 different subunits. And these are arranged as this really large, hollow, spherical shell. And so what happens is iron ions are transported into this shell as ferrous iron. So there's some spaces between these various subunits. And then it's oxidized to ferric iron. Ferric iron has very low solubility. It forms these clusters and crystals with phosphate. And this prevents it, it's a very good storage form for the iron. And it also prevents it from leaving this storage cage. And so once it moves in, so it moves in as ferrous, it's oxidized to ferric iron, which is a very good storage form. And you can pack about 45 iron ions inside one of these ferritin storage compartments, which is a lot. So it serves two functions. It serves as an iron reservoir, so a buffer to store iron and to release iron when we need it. And it's a really nice means to safely store the iron, since it's less likely to contribute to any production of reactive oxygen species once it's safely sequestered in this compartment. safe storage compartment. Okay, so we can either use it, we can store it in ferritin, storage compartment, or we can export it for use by the rest of our body. Right? So we're still in the enterocyte, and we can export it out into circulation. And this is going to be mediated by ferroportin. And so this is easy to remember because it's ferro, iron, important, the transporter. And this is the sole iron exporter. Okay. So it can export both ferrous iron and zinc. And it's an integral membrane transporter expressed on this basolateral membrane of the enterocyte. And it will be important to note that it undergoes multiple mechanisms of regulation, both in terms of its expression and its ability to transport the iron. We'll talk about that in more detail later. Once it gets out into the, once it's transported across the membrane by ferroportin, it's transported as ferrous iron, so Fe2+. And it's picked up by this transporter transferrin. And this is going to mediate transport. It carries the iron through our circulation to various tissues. But transferrin only binds to the ferric form. So there's a lot of conversion here between the ferrous and the ferric forms. So it only binds the ferric form of iron. So we have to oxidize it back to ferric iron. And there are two proteins that do this. Ceruloplasmin, plasminin, which is secreted by the liver. It's a soluble protein. So it's going to convert ferrous to ferric iron. And then there's this other one, this hephaestin, which is a related protein and is an integral membrane protein that's on the basolateral membrane of the enterocyte. And they have very similar activities. They're homologs of each other. But this one's an integral membrane protein. And it's going to convert ferrous iron also to ferric iron. And so this is important because this allows it to be packaged onto transferrin for transport. And this also keeps levels of ferrous iron very low on this side of the basolateral membrane of the enterocyte and supports efficient efflux from continued transport across that membrane. And so this transferrin molecule protein, it's a soluble protein in blood, and it can bind two iron ions, but they have to be in that ferric form, so it doesn't bind ferrous. And so I added this slide, so it isn't on the test or anything, but this might help you remember the names. So this hephaestin, right? So we talked about these two, ceruloplasmin and hephaestin. This hephaestin was an enzyme actually discovered here in this department by Chris Volpe in the late 90s. And he recently moved to Florida, but he discovered this when he was a professor here on the 4th floor. And so he discovered this, and he named it after Hephaestus, who's the Greek god of blacksmithing, metals, and fire. So it's kind of a neat name and a way to remember it. So it kind of deviated from most of the names which had fer in it, ferroportin, ferritin. But at least it has this cool reference to the Greek god of metal. Okay, so Hephaestin comes from Hephaestus, this Greek god of metal and blacksmiths. And it was discovered here at Cal, so go Bears. Okay. So we've moved now from the enterocyte out of the enterocyte through this ferroportin. Yes, ceruloplasmin hephaestin, which convert the ferrous iron to ferric iron. This allows it to now associate with transferrin for transport through our body, through our blood, through our circulation. And this is the crystal structure of transferrin. It's a dimeric protein, and you can see it here coordinating two different iron molecules, both in the ferric state. Okay. It's synthesized in the liver and secreted, so it's a soluble protein. As we've mentioned already, we only have a very tiny amount of iron that's circulating through our blood, so around 4 mg. So a very small amount of total blood is actually associating with transferrin at any one time, but there's a lot of flux in this compartment. So normally transferrin in this compartment is about 30% saturated with iron, so there's molecules of transferrin that are free and able to pick up new iron. Under 16% would be indicative of iron deficiency. If you have over 45%, this is indicative of iron overload. So too much iron. And when you start to have something like this, over 45%, you start to get free iron accumulating, which is never a good thing. So let's put all these together in terms of absorption of iron. So in the gut lumen, we have our iron in these various forms, the ferric, ferrous, and heme form. These are going to be taken up by different types of transporters. The ferric form is highly insoluble, and it can't be taken up directly by our cells. It has to be first reduced to ferrous, right, by our duodenal cytochrome E, DCYP. This can then be taken up by DMT1. The heme can be taken up directly, and iron released by breakdown of that heme by heme oxygenase. So we get this labile or transient iron pool. There's a few different fates: storage or utilization, or efflux to our body, right? So it goes out through ferroportin, it's oxidized to ferric iron by based in ceruloplasm and loaded onto transferrin. So are there any questions about this process? Okay, let's take a short break. So 5 minutes. Circulation and uptake through transferrin, and then move on to some of these regulatory mechanisms. So this is an illustration of the uptake of transferrin, which is bound to the ferric iron. And so we'll just walk through this. So the first step, we have ferric iron, and it binds 2 irons. So this is just showing the 2 irons bound to transferrin. It's moving through circulation, right, through our blood. This ferric iron can bind to a transferrin receptor, which is presented on the cell surface of a variety of different cell types. And then this can initiate the endocytosis through clathrin pits. Right? So if you recall clathrin-mediated endocytosis, we have recruitment of clathrin, we have the endocytic pit forming, and we have diamond pinching off a vesicle. So we have this clathrin-coated vesicle forming and pinching off into the cytoplasm. This is then going, so now we have this vesicle, and it contains our transferrin receptor bound to transferrin, which has those 2 irons. It'll also contain some DMT1, the transporter that transports ferrous iron. Okay, and then as this matures, so it's going to fuse with endosomes, and we'll have protons being pumped into the core of the endosome and acidifying the lumen of the endosome. So the pH of the endosome begins to change. This is really common for endosomes. A lot of times you'll have the endosome mature all the way to like a lysosome, where it's going to be very acidic for breakdown of the contents. Okay, and so we have an acidified endosome, so the pH around 5.5. At this pH, the iron dissociates from the transferrin and then is reduced and can be transported out through DMT1 into the cell. So we have release of the iron and transport out into the cell, into the cytoplasm. Okay, and the transferrin receptor and the transferrin remain in the endosome, and they remain bound to each other until they merge back with the plasma membrane. So the transferrin receptor and transferrin are both recycled. So we have those merge back with the plasma membrane, and under this pH, it releases transferrin, and this can re-enter the circulation and bind.
Did this transfer receptor bind to?
It binds a dimeric transferrin. So transferrin is usually a dimer, and each one of those is coordinating a single iron, and then it binds one of these, a single dimeric transferrin. And actually, I think the next slide has an image of this. So this would be an example of transferrin. So it's a dimer, so 2 monomers with a core of ferric iron, each present in each of these. And then there's a pocket formed by this transferrin receptor. Okay. This can fit right into the pocket and induce endocytosis of the transferrin receptor complex. Right. So the steps being,
Sorry, going back to the last slide, is Does the endosome just describe a matured clotting vesicle?
The endosome, so it can. So it gets a little tricky. So sometimes these vesicles will be quite small, and sometimes usually what will happen is that vesicle won't just mature on its own. It usually fuses with a lot of other vesicles to form these larger endosomes, and these will form into these acidified compartments and into recycling endosomes. So there are a lot of triage decisions that happen there. It's really interesting, actually, because there's this triage that sends some cargo towards the lysosome for degradation, and then some cargo for recycling.
So it's not shown in this picture, but pretty much the endosome is much larger than its initial
Than the
vesicle.
initial vesicle.
Okay.
Yeah, likely. Very likely, yes. Okay, so these steps are just laid out here. We have transferrin binding the receptor. This undergoes endocytosis. There's acidification and maturation of the endosome, which will trigger the release of iron from transferrin. This iron can then be transported into the cytoplasm, and the empty transferrin receptor recycled back to the plasma membrane for reuse. Any questions about that? Yeah.
Go back to the same pictures.
So,
So transparent is usually a dimer, and in each part of the dimer binds one iron, but also is the receptor used to dimerize to induce
yeah, the receptor is also an oligomer. So you can see that sort of represented here. They don't have crystal structures of the transmembrane regions, but you see it has two oligomerized halves.
So basically every time it's 4 iron.
No. So it's a dimer, but each of these
It's one.
has a single ferric ion.
Do this both of those?
Oh, here.
It looks like 4.
I will double check on that. I don't know if this is accurate, this illustration, but I'll check on that. That's a good question. At least from the crystal structure, it looks like it would only bind 2, but I'll double check to see if it could bind to more than that.
And from the previous diagram, the DMT one also gets in the vesicle. What's
Yeah.
the purpose of that?
It's to transport the iron out of
okay.
the vesicle. Yeah, so it has to have an exit. It's good eyes. I didn't even realize that.
And that's the whole thing to do something like that.
Yeah, no, good question. I will double check on that and let you know in the next lecture. I'm not sure about that. So not surprisingly, iron levels in the body are subject to multiple mechanisms of regulation. So one is at the transcript level. So they're what's referred to as iron response elements. So these form this very characteristic hairpin or stem loop structure shown here. And these can be present either at the 5' end or the 3' end of the transcript. So this is an mRNA transcript, so we have a 5' end, a 3' end, and then we have this coding region. Right? So ribosomes are going to track along this transcript and code for particular proteins. And so we can have these hairpins, these stem loops, these iron response elements, either the 5' or 3' end, and they'll have different effects on the transcript and protein translation. And so proteins with iron response elements or transcripts that encode these proteins would be things like ferritin, ferroportin, and transferrin. And so all these things involved in iron metabolism. Okay, so this is recognized, this iron response element, this stem loop structure, is recognized by a particular protein. And so I referred to this kind of at the beginning. This is a protein that has one of these 4 iron, 4 sulfur clusters. So this is a representation of that protein in the iron-bound form. It has its closed conformation with this iron-sulfur cluster kind of contained within that closed conformation. But this iron-sulfur cluster is unstable, and periodically it will lose the iron and it has to be replaced. Okay? So under conditions of low iron, you'll have proteins that lack this iron sulfur cluster. And under these conditions, the protein adopts a different conformation. So it's a more open conformation, and it's able to bind the mRNA stem loop structure, this iron response element. So it's kind of a cool way of monitoring iron levels. So high iron levels, this protein is going to have that iron sulfur cluster. It's going to be in that closed conformation. Low iron levels, it loses this iron sulfur cluster, and will have more in this open conformation, which can bind these iron response elements. And so this will have various types of effects. So as I mentioned, under low cytoplasmic iron, this will increase IRE binding by these iron response protein, iron regulatory proteins. So you'll sometimes see these referred to as IRPs, iron regulatory proteins, sometimes as IRE binding proteins. They're the same thing. And either name is fine. So the IRP binds to the IREs, and this can affect either translation or the stability of the transcript. So if the IRE is at the 5' end of the transcript, it's going to prevent translation. And you can kind of think about this if you imagine like a ribosome tracking along this transcript. If there's a big IRP bound in IRE here at the beginning, it's probably going to get in the way of the ribosome reading the transcript. Okay? Whereas if there's a 3' end, that ribosome can read right through the whole coding kit. Right? So if it's a 5' end, this is going to prevent translation of the protein. So if an IRP binds to that, it'll reduce translation, you'll reduce protein levels. If it's a 3' IRE, this is going to increase mRNA stability. So this will have the opposite effect. So increase synthesis of the protein. So you have different effects if it's a 5' or a 3' IRE. So we'll show some examples of this. So here we show an example of 5' IRE. So it's at this left part, which will be the 5' end, and we have this IRE bound by an IRE binding protein. So if mRNA translation is stopped when iron is low, this is going to decrease protein levels. And so examples of proteins that have, or transcripts that have this are the transcripts that code for the proteins ferritin, ferroportin, or this aminoglutamate synthase, which is important for heme synthesis. We'll talk about this enzyme more when we talk about the synthesis of heme. Okay, and if you have an IRE at the 3' end of the mRNA and an IRE binding protein binds, it's going to stabilize the transcript, and this will lead to increased protein levels. Okay? And different transcripts have this 3' IRE. So one example is the transferrin receptor. That is a 3' IRE. Okay, so let's just break this down. So if we have low iron, we're going to reduce translation of transcripts that have a 5' IRE. So for example, ferritin will reduce cellular storage capacity, releasing iron. Kind of makes sense if you have low iron. Ferroportin, so if the cell detects it has low iron, it might decrease its export of iron, right? So it'll reduce the translation of ferroportin. And then this enzyme involved in heme synthesis, if it has really low iron, it's going to reduce the synthesis of heme because it doesn't have enough iron. Right? And then for mRNAs with a 3' IRE, these are going to be stabilized. So if it detects low iron, it's going to try and increase transferrin levels in the circulation, hopefully increase iron being brought to the cell for uptake to replace its low iron. So transferrin has this low 3' IRE, and when there's low levels of iron, it's going to bind. The IRE binding protein will bind, and you'll have stabilization of the transcript and increased expression. Right? And the opposite happens when there's high iron. So if there's high iron, that IRBP is going to have that iron sulfur cluster. It'll be in the closed conformation. It won't be able to bind these IREs, the stem loops. So translation will no longer be repressed for those with the 5' IRE. So high iron, you're going to increase the translation of ferritin, so you're want to get an increased storage, right? You're going to increase ferroportin, so you're going to increase iron export from the cell, so it's detecting that it has high amounts of iron. And similarly, the mRNAs with the 3' IREs are going to start to be less stable, so they'll start to be degraded. So the transferrin receptor transcript will start to be degraded, and this will reduce expression. So the other mechanism for regulating, so that mechanism is at the transcript level, which is kind of unique. So we've talked about various ways in which receptors and transporters are regulated at the gene level, right, by transcription factors regulating the gene. And so this is kind of a really cool, unique way to regulate the transcript. And one nice thing about regulating the transcript is it can be faster than having to go back to regulate gene expression at the DNA level. So it can respond more quickly to these changes in iron concentrations. So another mechanism to regulate plasma iron is through this small protein or peptide hepcidin. And this is around 25 amino acids. It's secreted from the liver into the circulation. So it's synthesized and secreted by hepatocytes. And the levels of hepcidin are positively correlated with iron. So the more iron you have, the more hepcidin you have. The lower the iron in the plasma, the lower the hepcidin release. And it mostly circulates in a free state. And it regulates iron by controlling export of iron through ferroportin. So it directly binds to ferroportin, which is our sole iron exporter from cells, and it will induce the endocytosis of ferroportin and its lysosomal degradation. Okay? So if it's detecting high amounts of iron in the plasma, it wants to reduce this, right? It wants to reduce the levels of iron, so it's going to bind to the transporter that's pumping iron out into the plasma, and it will induce its lysosomal degradation. So we'll have decreased amounts of ferroportin on the cell surface and decreased efflux of iron from cells into the plasma. So this is going to be really important in terms of regulating that transient pool of plasma iron. Okay, so I think we made it through a little early. Are there any questions on the material? Okay, you're welcome to come talk.